Okay, we're going to go ahead and finish off our talk about meiosis and sexual reproduction. So let's go ahead and recap on what we talked about last time. We talked about meiosis and the different stages of meiosis. So remember, we have to have interphase, and then we have meiosis 1 and meiosis 2. So this is just a recap on what happens during each of them. Remember, the whole purpose of meiosis is to turn a germline cell, which has 46 chromosomes, so it's considered diploid, into gamete cells, which has half the number of chromosomes, so they are considered haploid. Remember, after meiosis 1, we have a reduction in chromosome number, and we're left with 2 non-identical daughter cells at that point. In meiosis 2, we're going to be able to produce 4 non-identical haploid daughter cells, which are going to become our eggs and our sperm. So, what causes or what allows for the genetic variation? So we did mention some in the last lecture. We're going to go ahead and get a little bit deeper in each of those. We talked about crossing over, independent assortment, but let's go ahead and also talk about random fertilization and mutations. So any kind of mutation that occurs can actually lead or allow for genetic variation of a new individual. Okay, and for the sexual reproduction portion of genetic variation, we did hit on crossing over and independent assortment the last lecture, but also if you think about random fertilization also allows for greater genetic variation as well. The reason being is 2 individuals can actually breed with another and actually create a new individual or allow for offspring, and there's nobody that actually basically says this individual can only reproduce with this individual. It's completely random fertilization of who or which 2 individuals come together to make a new offspring. So remember, we talked about crossing over, and it occurred in prophase 1 of meiosis 1. It is basically a process by which a chromosome and its homologous partner exchange that heritable information at the tips of their chromosomes or somewhere close to it. So for example, right here, we actually have these 2 different homologous chromosomes. So here, for example, again, this would be chromosome 1 from mom, this would be chromosome 1 from dad, but remember they got replicated during the S phase of interphase. So now what happens is that they actually overlap one another. Remember that overlapping where they're on top of one another kind of thing, that's actually called synapsis. And then the point at which they actually cross over with one another and exchange the actual genetic information, that's called the chiasmata point. So at the very end, when they're done crossing over, this will have a new endpoint over here, as well as a new genetic information, including all the genes on this one right here. So now let's go ahead and talk about independent assortment. Independent assortment is where homologous pairs of chromosomes orient or randomly line up at metaphase 1 of meiosis. Right here, you will see both the big blue chromosome and the little blue chromosome line up on the left side, and on the right side, we actually have the long red and then the little red. So this is what meiosis would look like if this lineup occurred. Very end, you're left with this combination and that combination. And this is not showing crossing over, this is just showing you independent assortment. Possibility number 2, though, remember, it's going to completely randomly assort where they want when they're going through metaphase. So right here, we actually don't have the 2 blues on one side, we actually have the big blue and the little red. Completely, they're going to separate completely independent of one another. So each nucleus is going to divide again in meiosis 2. Remember, we have 2 daughter cells, and then at the very end of meiosis 2, we end up with 4 unique daughter cells. So the human germ cells that are actually created, which are our gametes, have 23 chromosomes in them. Each time meiosis occurs, each of the 4 gametes that form have one of 8,388,608 possible combinations of homologous chromosomes. Reason being, all they did was put the 2 chromosomes times the 23 pairs. That's how they figured out all the different possible combinations that can occur. So as you can imagine, that's a huge amount of genetic variation that can occur in an individual. Let's go ahead and compare mitosis to meiosis. So these are nice diagrams. Right here is the parent cell. If you were going through mitosis, you would go this direction. If you're looking at meiosis, you're going to go this direction. Mitosis only has 1 round of PMAT. They both have to undergo interphase, especially the DNA replication, which is the S phase. So right here, it shows chromosomes have been replicated, once again, S phase. And now you can see there's 2 on each of these individuals right here. During mitosis, remember they have prophase, where nuclear envelope actually starts to break down. You also have the spindle microtubules start to form and go to opposite poles. You have a lot of stuff occurring during prophase, but there's no crossing over during mitosis. There is, however, crossing over that occurs during prophase 1. So it's very, very different. Right here, during metaphase of mitosis, you have all the sister chromatids line up on the actual metaphase plate. Over here on meiosis 1, you actually have the tetrads or pairs of chromosomes line up on the metaphase plate. So sister chromatids get separated during anaphase of mitosis, and you're left at the very end of the day with 2 daughter cells that are still diploid and look identical to the parent cell. If you guys compare it and look through each one of these, they have the exact same chromosomes that the 2 new daughter cells have. Meiosis, on the other hand, remember we're lined up here with our tetrads on our pairs of chromosomes on the metaphase plate in meiosis 1. Then they separate out, and we have full chromosomes go to each of the new poles and form 2 new unique daughter cells, because this daughter cell does not look anything like the parent. This one also does not look like anything like the parent, so that's why it's 2 non-identical daughter cells. Then those undergo meiosis 2 to further divide, and now they are left each with 3 chromosomes in each of the new unique haploid cells. And we say that they're haploid once again because there's 1, 2, 3 centromeres here, so 3 chromosomes. Over here there's 1, 2, 3, 4, 5, 6 centromeres, which means there's 6 chromosomes. So we actually got cut in half when we went through meiosis. Here's a nice little chart you can follow. Chromosome complement. Mitosis, we're in diploid. We start off diploid, we end up diploid. Meiosis, we start off diploid, end up haploid. Genetic variation, there's no genetic variation in mitosis. However, there is a lot of genetic variation due to crossing over and independent assortment in meiosis. There's only 1 division in mitosis, but there's actually 2 divisions in meiosis. And what divides during each division? So mitosis, since we only have 1 division, there's only that 1 part during anaphase where the sister chromatids get pulled apart. Whereas meiosis, we have 2 divisions. So the first division is actually separating those homologous chromosomes, or those tetrads get pulled apart. And then the second division is when we actually separate our sister chromatids. The cells that are involved in each mitosis, we have our body cells, our somatic cells, which are every cell besides your gametes. The actual meiosis cells that are involved in meiosis are going to be your gametes, which are known as your sex cells. And any kind of other characteristics that you should take note of or understand is, in particular in meiosis, you should understand what synapsis is, the chiasmata, what a tetrad is, and what crossing over and independent assortment are. So remember how I said the males actually form 4 gametes and they actually have 4 viable sperm cells? So at the end of meiosis, at the end of spermatogenesis is what it's called, you actually have 1, 2, 3, 4 unique sperm cells that have become haploid. Because look at the beginning, they're diploid, they become haploid. All of these 4 sperm cells are still viable and will be used to try to fertilize the egg. On the other hand, in oogenesis, you actually have a diploid that a woman starts off with, and look what happens. You can still follow along, it still ends up with 4 different individuals, 4 gametes, but only one of them will be the chosen one and later be fertilized to go ahead and make a baby or make an individual zygote. These other 3 become polar bodies and basically disintegrate into the actual woman. Now with anything, there are complications that can occur when things don't work properly because not everything's perfect. So during division of meiosis, there are things that can happen which does not allow perfect separation of those chromosomes from one another. Nondisjunction can happen during meiosis 1 when those tetrads are supposed to separate apart. Okay, so normally, remember these guys, the tetrads are supposed to be completely apart. Just like you see right here. This is how it should look, but this is nondisjunction. Nondisjunction is when basically those homologous chromosomes or sister chromatids fail to separate. So basically one will go with the other one when it shouldn't. The result of this is we just follow along the same process of meiosis. It keeps undergoing meiosis, but at the very end, if you look at this, remember, there's 4 chromosomes that we start off with. Remember, there's 4 centromeres. At the very end, we should have 2 in each of our cells, but instead, we actually have 3 right here. That's why it says N+1. We actually have 3 right here also. We actually have a reduction right here. We should basically have 2, but we have less than 1, so it's N-1, N-1. And a normal individual should have 23 chromosomes coming from mom, 23 coming from dad. But if you have extra chromosomes coming from one of those individuals, it can create a lot of abnormalities and lead to different syndromes, which we'll look at in just a second. On the other hand, we just looked at nondisjunction when it occurs during meiosis 1. But what if meiosis 1 happens and everything works great? Those tetrads are pulled apart from one another, and now the nondisjunction happens during meiosis 2. Let's look at those results. During meiosis 2, we actually have nondisjunction for these sister chromatids that's supposed to separate. So this is separating correctly, this is not. Both sister chromatids are going towards one side. The result of this nondisjunction during meiosis 2 is that we'll have 1 extra chromosome in one cell, we'll have a reduced number in the second cell, and 2 normal gametes that are created. If you compare these two, nondisjunction during meiosis 1 is actually worse because all 4 of the gametes that are created are wrong. All of them are going to either be missing a chromosome or have an extra chromosome. Over here, only 50% have issues, so you can still have the chances of these 2 being the ones that actually fertilize, let's say the egg, if these are sperm. These 2 individuals can actually create a normal individual, whereas these 2 would allow for some abnormalities to occur. Let's go ahead and take a look at those abnormalities that I'm talking about. So, Down syndrome. It actually allows for an individual to be born with a smaller head at birth. They have developmental issues, they have dental issues, smaller ears, decreased muscle tone, among a ton of other symptoms. Okay, the reason for that is nondisjunction. Down syndrome, which is also called trisomy 21, tri means 3, it means that an individual is born with 3 chromosomes on their pair 21 set. So instead of the 2 that you've seen in those karyotypes, they actually have 3 chromosomes, so an extra chromosome that got put into that chromosome 21 slot. That's how simple 1 extra chromosome can actually disrupt and create an individual to have abnormalities compared to what's considered a normal individual. Down syndrome is actually have a big correlation with the maternal age of the mother. So if we look at this chart right here, the graph, you can actually see the mother's age right down here, and right here would be the risk of having an individual with Down syndrome if you have a live birth, so if the individual survives during birth. Right over here, if you look at this, so at age 35, you do see an increase of individuals born, or the chances increase of individuals born with Down syndrome. After 40 years old, though, there's a huge incline of a probability that you'll have a child born with Down syndrome. The reason being is that women's microtubules, remember those ones that are actually supposed to grab on? Let me pretend. Let's say this is my chromosome. Remember, the microtubules are what's grabbing onto the kinetic core and pulling these guys apart. Well, as a woman ages, their microtubules might not be functioning correctly. So instead of this microtubule being able to come over and grab onto this chromosome, instead it's going to have some glitches, and then when they pull apart, it's not going to be able to do its job. That's why as women increase, they actually tell you it's a higher chance of having abnormalities or children born with abnormalities. Another abnormality that can arise if nondisjunction occurs is what's called Cre-du-Chat syndrome. Cre-du-Chat syndrome is associated with some nervous system abnormalities and identifiable physical features, which you can see in these pictures right over here. It results from the deletion of most of the small arm of chromosome 5. So basically, instead of 2 nice big structures on chromosome 5, you'll actually be left with one this small or one this size. Remember, they're supposed to be identical in size, length, genetic information, everything, but these guys are actually not. This actually results in an individual that has a characteristic high-pitched cry. It almost sounds like a cat. It's like meow, meow, but that's actually the infant crying. And I did include a really good YouTube video right here. You can click on and listen to it. Very interesting and very good to know. This is the link in case this video does not play. Another syndrome that might occur due to nondisjunction is what's called Klinefelter syndrome. And I actually have a picture right here so you can actually see what occurs or what the issue is. So remember, we're supposed to have one pair of chromosomes per chromosome set. So chromosome 1 should have 2 chromosomes, chromosome 2 should have 2 chromosomes, chromosome 3 should have 2 chromosomes. Remember, we talked about the actual sex chromosomes. There should be an X and an X or an X and a Y. Klinefelter syndrome is when they actually, an individual has resulted in 2 or more X chromosomes in males. So basically they still have an X, they still have a Y, but they have an extra X. So in this karyotype right here, you can see here's my 2 X's and here's my Y. So it's actually considered a male individual here, but they actually have symptoms such as weaker muscles, greater height, poor coordination, less body hair, and breasts that actually are able to develop due to the extra X chromosome there. Another syndrome due to nondisjunction is what's known as Turner's syndrome. Turner's syndrome, unlike Klinefelter syndrome, remember we had an extra sex chromosome. In Turner syndrome, we actually are missing a chromosome. So it's actually a female individual. They have an X and then they have nothing else. So right here in this karyotype, you can see there's an X and this is an absence of an X. There's actually no other sex chromosome. Remember, because we have to have a Y chromosome to make somebody a male. So this individual, just one X and then nothing else, no other sex chromosome, then they are considered a female. But this is once again called Turner syndrome. What happens with Turner syndrome is that people have developmental delays. The females have a shorter stature, so they're actually a lot shorter than normal. And they're infertile, which means they can't have kids. They actually fail to attain puberty, and they have cardiac abnormalities. So once again, we just talked about difference between meiosis and mitosis. We also talked about nondisjunction and the difference between when it occurs during meiosis 1 versus when it occurs in meiosis 2. And we also talked about different syndromes that can occur due to nondisjunction occurring in either of those phases.